To achieve reproducible cycles of cell suspension culture, growth characteristics of the cell suspension culture lines are analyzed by measuring two major parameters. One is the cell viability and the second is the cell density of the cell lines over time. In addition, contamination of foreign organisms in the culture is also checked regularly. Materials required for analyzing cell viability and cell density are samples of suspension culture cells from first week and third week of culture is taken in the Eppendorf tubes for determining cell density. FDA stained cells for accessing the cell viability is taken in an Eppendorf tube covered with aluminium foil. In addition, we require microscopic slide, micropipette, Petri plates, cover slip, chemocytometer, compound light microscope and fluorescent microscope. Methodology. Initially, cells are observed under microscope to verify cell propagation and to detect for presence of any contamination. To observe the cells, a clean microscopic slide is taken, labeled appropriately, and samples of the cells for the analysis is taken on the microscopic slide. Place a cover slip and observe the cells under microscope. Observe the cells under light microscope. So when observing the cells in the light microscope, in 40x magnification, in the 0th day culture, we were able to see very few cells. There were single cells and cell aggregates also observed in the day 0 cells. In the week 3 culture, mostly the cells were found as cell aggregates when observed under microscope. Cell viability for the suspension culture is examined by FDA staining assay. Here, 1 ml of the suspension culture cells were mixed with equal volume of FDA stain. Cells mixed with the FDA stain was incubated in dark at 25 degrees centigrade for 5 minutes and the FDA stained cells were observed under fluorescent microscope. For the microscopic analysis and cell counting, samples of FDA stained cells were taken on a clean glass slide, and for cell counting on chemocytometer, placed cover slip and the slides were covered with aluminium foil and taken for observation under fluorescent microscope, which is present in research facility 3 in bioengineering block. So using this microscope, fluorescent microscope, we analyze the FDA stained cells. The FDA stained cells taken on the microscopic slide is placed on the fluorescent microscope and the images are captured using Q-Capture software. The images are captured and processed using the software. Viable cells exhibited bright yellow-green fluorescence. The individual cells and the cell clusters, viable individual cells and viable cell clusters exhibited bright yellow-green fluorescence. It is essential to maintain maximum cell density in the suspension culture for an effective productive process. Direct cell counting under microscope is one simple method of estimating the cell density in the suspension culture. It gives reliable count on the number of actively dividing viable cells in the culture. Cell counting for determining cell density is performed using hemocytometer. In a hemocytometer, cover slip is placed. The cells are filled in the counting chamber and initially observe the slide under low magnification to locate the square field. Each chamber is divided into grid patterns and we count the cells in the 4 large squares, which is again divided into 16 boxes. So we count the cells in the large 4 corners, 16 boxes in the large squares. So we count the cells in the 16 boxes. And when we count the cells which are present in the box and leave out the cells which are present on the line. Suspension cells, when observed under a microscope, had a heterogeneous population of cells, single cells, elongated cells, and cell aggregates. And cells, when the cells are observed under fluorescent microscope, the live cells are fluorescent green and the dead cells are unstained. So we take a count of both live and the dead cells and calculate the percentage cell viability using the formula total live cell count by total cell count into 100. When monitoring the cell suspension, once the cell density and the percentage of viable cells are known, the suspension cells are proceeded for subculturing of the cells into fresh medium.